A very good morning. Today we are going to deal up with the topic that is called as transposable elements in bacteria. Now, the transposable elements are basically the DNA molecules that are lying in the genome that move from one part to the another by means of an enzyme that we call as a transposase. And there is requirement of the specialized sequences that are called as the terminal inverted repeats. Now, in case of the bacteria, we say that these transposable elements or the jumping genes are generally categorized into three basic types. The first being the insertional sequences. Now, these insertional sequences, they are the simplest transposable elements that are present in the bacteria and are comprising of the genes that are required for its transposition only. They are generally of 768 to 2500 nucleotide pair long and involve the transposase enzyme only. Besides it, they have the terminal inverted repeats, that are the inverted sequences of 9 to 14 nucleotide pairs. The common example is the IS50 element, which is having a length of approximately 1533 nucleotide pairs. An important characteristic feature of this insertion sequence is that wherever it gets incorporated or this fragment gets incorporated, it carries out the duplication at the site of incorporation, which is represented by the target site duplications, that is TSDs. Now, the second important transposable element in bacteria are called as the composite transposons. Now, the composite transposons are the transposons which are made up of two insertion sequences elements that lie near one another, and the region in between them can be easily transposed by means of the joint action of the flanking regions or the flanking elements. So we say that the two IS elements are there, considering the example as IS50L and IS50R of 1533 nucleotide pair long, and in between them there are the three important genes that are kanamycin resistant gene, bleomycin resistant gene, and streptomycin resistant gene. Now, the characteristic feature of this is that it provides resistance against certain antibiotics, and they are more complex or they are more advanced as compared to that of the insertional sequences. The commonest example is that of the TN5, which is 5700 nucleotide long. Now, the third important type of transposition or transposable elements are the non-composite or complex transposons. They are generally characterized by the presence of 38 nucleotide pair inverted repeats that are lying on their ends, and these are the simple inverted repeats. Besides it, there is a presence of various other genes in between them, so that's why they're called as the non-composite or the complex transposons, like that of the TNPA, which encodes for the transposase. Then we have TNPR, which encodes for resolvase, and we have the BLA gene, which encodes for the beta-lactamase or ampicillin resistance. Now, besides providing the resistance gene and their transposition, it has a rest site to which the transposase enzyme act and carry out the process of transposition. The commonest example is that of the TN3, which has a length of 4957 nucleotide pair and is one of the most important complex transposons. Now, this is all about the transposable elements in bacteria. Thank you.